should be most of the team who are here. Just checking that the live stream is working. And that is looking good. Fantastic. In that case, I'm very happy to get us started and super excited for Sooyeon Ji to join us from OHBM this afternoon for you, this lunchtime for us. She is currently an assistant professor for computer engineering at Hankook University and was previously a postdoc in Young Lee's lab for imaging science and technology at the Department of Electrical and Computer Engineering at Seoul National University, and has a background, I guess, in electrical and computer engineering also from Seoul. The floor is yours. Take it away. Super excited to have you. Thank you for joining.
Oh, it says that I don't have the permissions to.
Correct. We
Yeah.
set you up before and then took a break. So
Yeah.
let me just make you co-host again. And now you should be able to share your screen.
Great. Okay. I hope everybody had a lovely lunch, and thank you very much for the introduction and also for the wonderful opportunity. The main focus of my talk today is about magnetic susceptibility source separation for myelin imaging, as the title exactly says. So I will first go through the underlying physics of susceptibility source separation and how this method can and is being investigated as an indirect measure of myelin. So as many of you are already familiar of, myelin is a fatty substance that wraps around nerve fibers, which functions like insulation on electrical wires. And this insulation allows for saltatory conduction, which enables nerve impulses to jump between gaps in the myelin sheath. And this in turn affects the conduction velocity, ensuring efficient communication within the brain and spinal cord. So understanding myelin is especially important when considering demyelinating diseases such as multiple sclerosis or neuromyelitis optica disorder syndromes. And these conditions occur when myelin is damaged, leading to impaired neurofunction. And additionally, myelin also plays a key role in development and aging, and as well as brain plasticity. So imaging myelin can provide valuable insights into both normal brain function and also various neurological conditions. And myelin's unique properties contribute to various MRI contrasts, including T1-weighted imaging, T2-weighted imaging, FLAIR images, diffusion tensor imaging, magnetization transfer, and so on and so forth. So each of these imaging techniques may shed light on the structure and distribution of myelin. And susceptibility source separation, which is the main topic of today's presentation, is a method to image magnetic susceptibility, which is also really closely related to myelin itself. So in the human body, multiple sources contribute to magnetic susceptibility. And for example, the two main sources of susceptibility in the brain is the paramagnetic iron and diamagnetic myelin. Calcium and fibrosis are both diamagnetic, while oxyhemoglobin is diamagnetic and deoxyhemoglobin is paramagnetic. So to be particular, iron and myelin are known to be closely related to neurodegenerative diseases, and the amount of the two substances would simultaneously change during the progress of diseases. And therefore, quantifying or assessing the distribution of these substances would potentially provide valuable information about the diseases. So before moving further forward, I'd like to briefly review what magnetic susceptibility is and how it affects MRI. So magnetic susceptibility is a property of a material that describes how much magnetization it acquires when it's exposed to an external magnetic field. So a paramagnetic material, as illustrated in the middle panel, would enhance the magnetic field, aligning with it and then effectively reinforcing its strength. And conversely, diamagnetic materials, which is shown on the rightmost side of the panel, produces a magnetization that opposes the external field and slightly weakening the overall magnetic field. So in MRI, these changes in the magnetic field would directly affect the resonance frequency of nearby water protons and resulting in local frequency shifts. So these frequency shifts would provide crucial contrasts in susceptibility-based MRI techniques, and I will explain them in details from this slide. So there are multiple susceptibility-related contrasts in MRI, and I would like to describe them by dividing them into two different groups, which are magnitude-related contrasts and phase-related contrasts. So the most familiar one is the T2 star-weighted magnitude images, which are often observed using gradient echo imaging. And this is a magnitude-related susceptibility contrast. And this T2 star weighting can be quantified as the R2 star relaxation rate or T2 star time constant values. The phase of the complex valued GRE images also reflect the susceptibility effects. And using these kind of phase images, we are able to quantify the magnetic susceptibility values by using quantitative susceptibility mapping techniques. So from now on, I will go into more details about how these susceptibility sources would affect the magnitude and phase of the GRE images. So the brain microstructure that contains magnetic susceptibility, sort like iron or myelin, is usually much smaller than the MRI voxel size. So the rectangle here, shown here, is actually the MRI voxel, and these small spheres inside it, they are the microstructure with susceptibility. So while the sources are denoted as spheres for simplicity, the real microstructures can be much more complicated, such as in cylindrical myelin. But I will just assume that they are simple spheres from now on. So when a large main magnetic field, B0 field, is applied, the susceptibility sources induces microscopic field fluctuations around them, such as this. And this in turn causes field inhomogeneity inside the voxels. And the field inhomogeneities would consequently lead to spin dephasing, as can be seen here. And the dephasing spins would
we lose the audio?
We can't hear you right now. That's weird. Nothing had changed. It's just gone.
Yeah. What? I don't know what
Time
happened.
for PowerPoint karaoke. Who wants to have a go at telling the story now?
Is something wrong, or is it?
Oh, we can hear you
No,
again.
it worked. Now it worked.
No, it works. So where did I lose you then? Okay,
Just
so
after the spin dephasing.
Okay. So, well, this kind of local magnetic field inhomogeneities would result in spin dephasing, and that would also result in an accelerated magnitude decay inside that particular voxels. And this kind of accelerated magnitude decay can be characterized by the R2 star values, or the difference between the R2 star and R2 values, which is the R2 prime values. So here we can conclude that if we have a susceptibility source, and if the susceptibility source concentration increases, the R2 star value would also increase. And this can also be extended to cases with various susceptibility sources. So the existence of susceptibility sources would result in accelerated dephasing regardless of whether the sources are paramagnetic or diamagnetic. So in the case of mixed susceptibility sources here, the two types of sources would jointly accelerate the signal decay, as can be seen on the right figure here. So this means that both paramagnetic iron and diamagnetic myelin would increase R2 star and appear dark in T2 star weighted images. And this characteristic is really well observed in this T2 star weighted image on the left side, where the deep gray matter with abundant iron and the optical nerve with high myelin contents both appear dark. So now I will like to move on to the susceptibility effect on the GRE phase. So we will assume that V0 field is applied to a paramagnetic object here. And then we will now zoom into the voxels inside the paramagnetic objects. We can recall that the susceptibility effects on magnitude came from the intravoxel field inhomogeneity, which would be localized inside this voxel. Phase contrast, on the other hand, is determined by the macroscopic intervoxel interaction. So this means that the phase contrast of a single voxel is affected by the surrounding voxels. So these kind of macroscopic magnetic field components induced by each of the voxels are summed up, resulting in a magnetic field generated around the object like this. This magnetic field perturbation results in local resonance frequency change across and around the object with magnetic susceptibility, consequently inducing phase contrast. So to be precise in mathematical terms, the field map induced around the object can be modeled as convolving the susceptibility distribution map with a dipole kernel shaped like this. So in case of a diamagnetic object, the physics are exactly the same, but the sign of the induced magnetic field is opposite like this. So the frequency shift would be exactly opposite from the paramagnetic object to the diamagnetic object. So therefore, in case of the phase contrast, white matter with abundant diamagnetic myelin would have opposite contrast compared to the deep gray matter with abundant paramagnetic iron. So because the local phase or local field map is susceptibility map convolved with the dipole kernel, the local field map can be acquired by convolving a dipole kernel to the quantitative susceptibility map here. But the quantitative susceptibility map is what we want, and the local field map is what we can measure. So we are able to reconstruct the QSM map from local field map by deconvolving the dipole kernel from the local field map. So the procedure of acquiring these kind of quantitative susceptibility maps from MR phase information is known as quantitative susceptibility mapping, or QSM. So there are several pre-processing steps that we would go through in order to acquire the local field map from the actual MRI phase images, such as phase unwrapping, echo combination, background field removal, and so on and so forth. But I will not go into detail about this here. And instead, if you are interested, I recommend you to have a look at the QSM consensus paper. Okay, so to summarize the effect of magnetic susceptibility on the phase, paramagnetic iron and diamagnetic myelin display opposite contrasts on phase, and the phase information can be utilized to actually quantify the susceptibility values. Well, however, one drawback of QSM is that it can't distinguish between the paramagnetic and diamagnetic susceptibility sources that coexist inside a voxel. For instance, the QSM value inside a single voxel will increase when the amount of iron increases or the myelin decreases inside that single voxel. So this can be problematic in lesions where the amount of iron and myelin simultaneously change. For instance, let's look at this MS lesion on the right side of right side of the slide here. So in the flare image, we can observe this MS lesion here. However, this lesion is obscured and not visible in the QSM map on the right side of the slide. This lesion actually had demyelination and also decreased iron content compared to the surrounding normal appearing white matter. However, the amount of demyelination and iron decrease was canceled out in this QSM map. The goal of susceptibility source separation is to disentangle the effect of paramagnetic and diamagnetic susceptibility sources that coexist inside a single voxel by separately quantifying them. We use additional information and or biophysical modeling to do this. So this is the same lesion from the previous slide. Now we are able to observe by using susceptibility source separation that the paramagnetic susceptibility, which reflects iron information, and the diamagnetic susceptibility, which represents the myelin information, have both decreased inside the lesion here compared to the surrounding tissue. So multiple susceptibility source separation methods have been proposed and developed, but today I will focus on the Chi separation model, which was originally proposed by Shin et al. At Dr. Jong Lee's lab. So in the Chi separation model, we utilize the magnitude information along with the phase information in order to separate the two sources. So we have previously discussed that the paramagnetic and diamagnetic susceptibility sources, such as iron and myelin, both increase the R2 star or the R2 prime relaxation rate when they coexist inside a voxel, like this. So they both accelerate the signal decay, so they both increase the relaxation rate. On the other hand, QSM value would increase when the paramagnetic source increases or when the diamagnetic source decreases. So by using these two informations together, we are able to separately quantify the two sources. So this is the final model of Chi separation. So the red box here indicates the magnitude domain model, and the blue box here represents the phase domain model. By assuming a static dephasing region and homogeneous isotropic spherical susceptibility, we can say that R2 prime is proportional to the absolute sum of chi para, which is the paramagnetic susceptibility, and chi dia, which is the diamagnetic susceptibility. The local field map is the signed sum of susceptibility values convolved by the magnetic dipole kernel pattern. So by combining these two models together, we can obtain the objective of the Chi separation model. Here, R2 prime values and the delta F, which is the local frequency shift map, are the parameters that we measure and quantify using the MRI machine. And the chi para and chi dia are the paramagnetic and diamagnetic susceptibility sources that we would like to quantify. And due to ill-posedness of the dipole deconvolution procedure, one may utilize optional regularization as proposed in previous QSM-related studies. So this slide shows the entire Chi separation pipeline. So for Chi separation, or maybe for the original version of Chi separation, we need two different data sets of the same subject. So the first one is the multi-echo GRE data, and the second one is the multi-echo spin echo data. The local frequency shift maps and R2 star maps can be measured from the multi-echo GRE data, and also R2 maps can be measured from the multi-echo spin echo data. And finally, we can measure the R2 prime value using the R2 and R2 star maps by subtracting R2 from R2 prime, R2 star. So Chi separation is then performed by solving the minimization problem defined in the earlier slide using the frequency shift map and the R2 prime map. Okay, so now I would like to move on to the validation of the Chi separation method. So the Chi separation method was first validated using computer simulations. So assuming spherical paramagnetic susceptibility with varying spherical paramagnetic susceptibility sources with varying absolute values were assigned in the first row. And for the second row, the diamagnetic susceptibility values were assigned. And the third row was assigned both para and diamagnetic susceptibility sources. And MRI signal from each voxel were simulated using Monte Carlo simulations, and the simulated images were then utilized to quantify the paramagnetic maps and the diamagnetic susceptibility maps using the chi separation model. So in this Monte Carlo simulation validation, we looked for the agreement between the assigned susceptibility values and the actual reconstructed susceptibility values. And we could see that they are in good correspondence when they were single sources or when they are coexisting sources. And the method was further validated by using a phantom experiment. So susceptibility phantom was constructed using iron oxide as the paramagnetic source and calcium carbonate as the diamagnetic source. So similar to the computer simulation, on the first and second row, single positive or negative sources were inserted with varying concentration, whereas in the third row, the two sources were mixed together in a agarose phantom. Here we looked for the agreements between the reconstructed paramagnetic and diamagnetic susceptibility values from single source only rows and from mixed source rows. And we could find that they display good agreement with those from single sources. And it suggests that successful separation of the sources was able even when the both sources coexisted inside a voxel. When an ex vivo brain specimen containing the visual cortex was observed, chi separation maps here corresponded well with the distribution of iron and myelin. Iron map was obtained using a laser ablation study, and the myelin map was obtained using myelin staining. So in particular, we observed that the line of Gennari, depicted as yellow arrows here, which is known to have both high iron and myelin content, displays high intensity in both para and diamagnetic susceptibility maps. So we can say here that the method is reliable for separating between the paramagnetic and diamagnetic susceptibility sources, even when they are coexisting inside a single box. So after that, we conducted further comparison focusing exactly on the diamagnetic susceptibility map and the myelin map by ROI analysis and depth-wise cortical profiling. So first, we conducted a linear regression analysis between the ROI averaged chi-dia and ROI averaged optical density from Luxol fast blue staining. So this analysis revealed a good correlation between the two measures. And also we did a cortical profiling at the primary visual cortex. And the cortical profile matched well between the two measures. So the red line is the cortical profile we obtained from optical density in Luxol fast blue staining. And the blue line is the cortical profile that we obtained using the chi-dia maps. So this one is the chi-dia map, and this is the optical density map. And we can see that the two measures are really, the two measures will be matched well together with similar peaking points. And furthermore, when an ex vivo human thalamus specimen acquired at high resolution of 350 micrometer isotropic resolution was observed, it revealed really exquisite details of the myelin distribution inside the thalamus. And it was really consistent with the myelin histology map here. However, we must still note that this particular acquisition consisted of like 8 different orientation scans with scan time ranging over 10 hours. So this is not really a practical method, but still it showed that it is able to display this kind of myelin maps inside the human thalamus that was really close to that what we can observe in the histology maps. So when we observed this kind of maps in vivo, the in vivo para and diamagnetic susceptibility maps displayed high correspondence with iron and myelin stains from the literature. So in the deep gray matter of the in vivo data, Chi separation was able to reveal details of the brain anatomy that matches, The Chi separation revealed details of the brain anatomy, such as the globus pallidus and pulvinar in the paramagnetic map, and anterior limb and posterior limb of the internal capsule in the diamagnetic maps. Still, we have some limitations in image resolution compared to the previous slide because it was in vivo scans with scan time limitations. Moving on to the applications of the Chi separation, I think one of the interesting application of this is creating a normative template of paramagnetic and diamagnetic susceptibility maps. So these are several selected slices of the Chi separation atlas, which was presented in NMR and Biomedicine in 2024. And they highlight well-known iron-rich areas and also some myelin structures. And these kind of normative diamagnetic and paramagnetic maps potentially provides a reference for comparison in clinical and research settings. The normative diamagnetic susceptibility maps were then compared to the normative myelin water fraction maps, which is a more conventional approach of indirectly measuring myelin using. So when we compare them, so the upper row here is the Chi-dia map, and the lower row here is the myelin water fraction map. And I must note that these atlases were both masked using the white matter masks. So the two maps display similar characteristics, and when we observed the RI values and compared the RI values between the two different measures, the diamagnetic map showed high correlation with the myelin water fraction, suggesting that the method is effective for myelin measurement. Another application of Chi separation is the visualization of the cytoarchitecture related to iron and myelin. So in the middle panel here, this is one of the really well-designed studies that show distinct laminar distribution of myelin and iron from the cortical gray matter to the deep white matter. So here, right here, the iron, depicted as green line, has a peak right after the gray matter and white matter boundary, while myelin has a peak in further deep white matter. So when the laminar profiles of the paramagnetic and diamagnetic susceptibility is observed, we can see similar peaking points here. So the blue line here is the Chi-dia, and the red line here is the Chi-para. We can see that the paramagnetic susceptibility peaks near the superficial white matter, whereas the diamagnetic susceptibility peaks far into the deep superficial white matter. On the other hand, we could see that the QSM values, which shows the collective effect of the paramagnetic and diamagnetic susceptibility, has a peaking point inside the cortex. Has a peaking point inside the cortex. Not the gray one, the purple one. So it peaks inside the cortex. And it suggests that the decrease of the negative susceptibility can shift the peak of the QSM to the earlier point into the cortex. So Chi separation may be useful for studying diseases such as multiple sclerosis, where iron and myelin would change simultaneously inside a single voxel or inside the lesions. So these two are two lesions that were displayed in the original work. And the upper row demonstrates a lesion with paramagnetic rim, whereas the lower row displays a lesion that appears iso-intense in the conventional QSM, but appears hypo-intense compared to the surrounding normal appearing white matter in both the paramagnetic and the diamagnetic susceptibility maps. So the second row here, it suggests that both myelin and iron was decreased inside this lesion, which led to a iso-intense contrast in the conventional QSM. So like this susceptibility source separation may provide better insights into the disease progression compared to conventional QSM measures. So the next study I'd like to talk about observed 20 relapsing remitting MS subjects with a total of 183 lesions. And the lesions were observed twice within an average of 14.4 months interval using conventional QSM, R2-star, and also chi-separation maps. The lesions were first divided into 4 different groups based on their R2-star and QSM value changes between baseline and follow-up. So I'm not sure if you are able to see this in details, but so this group 1 is a group of lesions that had increased R2-star and increased QSM values. And the second group here is a group of lesions with decreased R2-star and increased QSM values, and so on like that. Using the chi-paramagnetic and chi-diamagnetic values, the 4 group could be subdivided by the paramagnetic and diamagnetic value changes in each lesion. So, for example, let's look at this first group here with higher increased R2-star and QSM at follow-up. So the predominant source of the increase in R2-star and QSM was the increase of paramagnetic susceptibility, which can be interpreted as iron accumulation. But by using chi-separation, this could be further classified into lesions with iron accumulation and remyelination, or iron accumulation and a slight demyelinations. Like this, susceptibility source separation can provide information about the separate evolution of paramagnetic iron and diamagnetic myelin throughout the disease progression. And this may offer additional insights compared to R2-star and QSM alone. The next study, this study is a more recent study that observes the changes in myelin within an MS lesion over time using longitudinal chi-separation imaging. So the authors simultaneously quantified the paramagnetic and diamagnetic susceptibility sources using chi-separation, and they demonstrated that a hyperparamagnetic sign in the early stage, which means that in that lesion, in the early stage of that lesion, the lesion had high iron accumulation. And they demonstrated that this hyperparamagnetic sign in the early stage is associated with impaired remyelination capability in MS lesions. So when a lesion had early HPS positive signal, the myelin content tended to decrease, whereas if the early lesion did not have hyperparamagnetic signal, the chi-dia value tend to increase, so meaning remyelination. Also, they were able to say that a lower number of lesions with hyperparamagnetic signal in the early stage was associated to clinical disability improvement in people with MS. And this is another study that assessed remyelination using chi-separation. They assessed a total of 108 MS patients, consisting of 5,030 lesions, and they assessed for remyelination in 2 years interval. And they found out that the amount of remyelination was associated with younger age, lower disability scores, and relapsing and remitting MS phenotypes at baseline, and that the increment of myelin correlated with clinical improvement measured by the EDSS score. Chi-separation is also being explored for distinguishing between diseases. So this study compared the MS patients and MS lesions to NMOSD lesions. So they observed 32 MS patients with 611 lesions and 15 NMOSD patients with 225 lesions. And they saw that the MS patients show higher proportion of hypodynamic lesions than those with NMOSD. And also they found out that the area under the receiver operating characteristic curve, which is the AUC curve of the proportion of hypodynamic lesion, was higher than that of the proportion of lesions with diamagnetic rim sign and the proportion of lesions with central vein sign, which suggests that chi separation may provide a better ways to distinguish between MS and NMOSD compared to the previously proposed signs such as the central vein sign or the paramagnetic rim sign. Well, while I have talked about the positive aspects and how the diamagnetic susceptibility map reflects myelin information until now, I must say that we must be really cautious as several artifacts, including those inside and around large vessels, can affect the map. So the red arrows on the left side display the vessel-related artifacts. The chi separation model itself does not account for the flow inside the vessels, and the flow-related displacement of vessel signals can result in large artifacts in the R2 star map. And the R2 star map or the R2 prime map is used to separately quantify the chi paramagnetic and chi diamagnetic maps in chi separation. So this kind of artifacts will propagate into the chi separation maps, and they are shown here as the red arrows. Also, we assume that R2 star or R2 prime is determined only by the intravoxel field inhomogeneity here. However, there are some non-local R2 star effects that arise near sources with larger susceptibility values, such as large veins. And this can generate artifacts pointed out by the green arrows here. And furthermore, if the assumptions of the models, such as the static dephasing region and homogeneous and isotropic susceptibility assumptions are violated, this also may induce some errors in the quantified susceptibility maps. Furthermore, the existence of other sources of susceptibility, such as calcification, hemoglobin, copper, and so on, would further complicate the direct interpretation of diamagnetic susceptibility map as myelin. So we must be really careful in interpreting the maps and always keep in mind that we are measuring the susceptibility and that they are not directly iron and myelin maps. While today's presentation mainly focused on chi separation and use of chi separation as myelin measures, there are other methods for susceptibility source separation that have and are being developed. So I would like to point out just some of them. So there are susceptibility source separation that work with GRE data only. So we, in the original chi separation model, we needed GRE and spin echo data, right? So we had to acquire two different data types. But there are models that work solely on GRE data, so it means reduced scan time. There are models developed for susceptibility source separation data acquired at 7 Tesla. And there are researches regarding more efficient data acquisition for this, or better models for the relaxometric constants and models that utilize multi-orientation data. So ongoing researches are continuing to improve and expand these techniques. And I believe that these more advanced models and advanced acquisition techniques would help in interpreting the diamagnetic map as a more robust measure of myelin. So to conclude the talk, I talked about iron and myelin, which are the two primary sources of susceptibility, and that they have opposite signs in the brain. And susceptibility source separation is a method that tries to separately quantify this paramagnetic and diamagnetic susceptibility using magnitude and phase domain models of the R2 prime and local field perturbations. Of course, we do have some assumptions such as static dephasing region, isotropic homogeneous susceptibility, and these assumptions, when they are not fulfilled, they may become a source of error in the susceptibility source separation results. The chi-separation methods may provide meaningful insights in neuroscience and neurodegenerative diseases, progression and diagnosis. So I've talked about the normative brain template using the chi-separation values and also MS lesion categorization, progression, and differentiation between the MS and NMOSD. However, I think that we need to be real careful when interpreting chi-dia and chi-para as myelin and iron because there are complications such as artifacts in the maps and the assumptions that we used inside the model, and also there are other sources of susceptibility inside the human brain. Okay, so I think that's all for what I have prepared for this talk, and I would really be happy to hear any questions from you. Thank you.
Thank you so much. Super interesting. Hands are up already, I can see. So Lilith or Lice, please take it away.
Hi, thank you so much for presenting it. Can you hear me?
Yeah, I can hear you.
Yeah. For me, it was a little bit complicated to understand, but I was wondering something connected with the last sentence of this presentation. So maybe I missed it. How do we know? I mean, this high separation is really great, and now you can have some more understanding. But at the same time, as you mentioned, how do we know exactly that it is myelin or iron? So why did you pick to say that we are separating myelin and iron, not like myelin and some other type of artifact? So why is it more likely to be iron than other type of source?
Susceptibility sources. Okay. Well, that's a really, really good question. And I would like to say that diamagnetic maps and paramagnetic maps, they are never myelin and iron themselves, but iron and myelin are the two primary sources of susceptibility inside the brain. So we assume that maybe, and I don't want to say assume, but like, yeah, we interpret that the diamagnetic map is related to myelin and paramagnetic map is related to iron. So let me give you an example. So if we have a calcification inside the deep gray matter, it would show a high diamagnetic susceptibility value in that area. But we don't say that that is myelin. It's just a high diamagnetic susceptibility value. But when we like think of a MS lesion, and if a MS lesion is progressing through time, and then we see like decrease in the diamagnetic susceptibility value, then maybe we may say that this might represent the decrease of myelin inside that particular lesion. So yeah, that's what we're doing right now. And again, diamagnetic maps are not really myelin maps. It's just a measure that we take to interpret the, yeah.
And are there maybe some preliminary ideas or, I don't know, something to manage to within each map to also divide into different sources, or is it something more, yeah, it's impossible to do it? So how can I say that this is iron, not calcium, for example? Not only say, okay, paramagnetic is it possible? Are there any thoughts about it?
Yeah, I do have some thoughts about it because I like obscured it a lot in this presentation, but the actual relationship between the R2 prime values and the susceptibility values, the actual constants between them, the DR constant. It differs when the size or the concentration of the susceptibility sources differ. So if we are able to measure or like disentangle this effect and measure the DR component, we may be able to separate between the different kinds of susceptibility sources. So just a moment, please. So here, we said that the R2 prime value is related to the susceptibility values with a constant here, right? But this actual constant would change if the susceptibility source distribution or the size of the source or the geometry of the source differs. So DR may differ by that. So if we are able to measure the DR and also separately for the paramagnetic and diamagnetic sources, then maybe we might be able to separate between different sources. So we may be able to say that, okay, so this voxel contains a pi power with this kind of DR. So maybe the main source of paramagnetic susceptibility in this voxel is iron, and this one is not iron. So I think that may be a way to go, but the point is that the point is that we only have two measures and we are quantifying two different values, right? So we would need more measures to do that.
Mhm.
Mhm.
Yeah, yeah. Okay. Thank you so much.
Thank you.
Maybe also has a question for you.
Yeah, thank you for this very informative, very interesting talk. So my question was about the so this source separation technique. So when I was wondering how the spatial co-localization or the geometry of iron and myelin would affect the fidelity of this source separation. For example, is there any specific tissue or region in the brain that the source separation is more successful compared to the other? Like what happens when there are like crossing fibers, white matter tracks with different orientations in the, how do you think it would, affect the success of this separation?
Okay, that is another really great question. Just a moment, please. So I'll just stay in this slide. So this model, chi separation model, was constructed with a lot of different assumptions, including the statically phasing region, homogeneous susceptibility. And here it says that it's under the homogeneous and isotropic susceptibility, right? So it doesn't account for the anisotropic susceptibility nor crossing fibers or something like that. So, as you know, like if you have like fiber orientation inside a voxel, then that would result in anisotropic susceptibility, right? And also, this model does not account for the frequency shift that results from the fiber orientation. So it says that it would only result in chi para plus chi dia convolved by the dipole pattern. But if we have like fibers with different orientations, we would have additional frequency components, right? So I think that this model would work when there are no fiber orientations right now, but I didn't mention here, but I'm not sure. Nah, I didn't include it in here because it's not a actual paper yet. In the ISMRM of last year, I think there was a model that accounted for the multiple different orientation fiber orientation data. So I believe that they utilized additional diffusion tensor imaging data to address the fiber orientation part of the frequency shift, and then they modeled it all together into the separation model. So I think using those kind of more advanced models, you would be able to get better separation in those kind of regions too. But for the high separation model of the original form, I think that it may not fail, but it may have some errors in those kind of areas.
Mm-hmm. And this is only tested for cortex or also for spinal cord?
The work was all done for brain,
Mm-hmm.
so not the spinal cord yet, but I think that is a really interesting way to go for this
Mm-hmm.
susceptibility source separation methods.
Yeah. Okay, thank you.
Thank you.
A lot of my questions have already been answered, but I still have at least two for you. So the first one is that we see the separation in the field where half the field is going towards higher field strength and the other half is going towards ultra low field strength. And I was wondering if you could comment on the impact of the field strength or the role in enabling an accurate separation, or if it doesn't matter.
Yeah, like every questions here are super interesting and they are so important questions. So I briefly mentioned that there are susceptibility source separation methods for data acquired at 7 Tesla, right? Well, the susceptibility value and the relationship between the local field map and the susceptibility map itself should not differ between different magnetic field strengths. However, the R2 star values or R2 values or R2 prime values, they differ when we use different field strengths. So we have to like overcome this gap between different R2 and R2 prime or R2 star values. And this particular work did that by using deep learning methods to like convert from the R2 prime map acquired at 7 Tesla to, R2 star map acquired at 7 Tesla to R2 star map acquired at 3 Tesla. But I mean, it solely depends on the data itself. So I believe that that particular method was trained on healthy controls. So I think we have to go more further into like using some more patient data and lesion data so that we can use them in other kind of field strengths.
Great. Two more questions, if I may.
Okay.
So you alluded to different methods being available, but one of the most prominent methods, as far as I can see in the field, is the T1-T2 ratio for myelin mapping.
Oh yeah, right.
And I was wondering how the susceptibility for separation compares to that method of the ratio.
Well, I think the T1 over T2 method, it's really useful because we always acquire T1 and always acquire T2, and we can just like get a ratio of those two and then see like myelin related contrasts. Well, one point is that method, T1 T2 ratio method, is not really quantitative, right? So you acquire different contrasts and then you just like divide them. So because they are not quantitative, the advantage of quantitative MRI is that you can really use them in longitudinal studies, right? If you just acquire like contrasts, the contrast value itself, signal value itself doesn't have a real meaning. So it's hard to use that in longitudinal studies. But susceptibility source separation is actually quantitative, although it may not actually mean myelin itself, but it is still quantitative, right? So we can use it in longitudinal studies. And I think that point would go same on the T1, T2 ratio methods, because like if we have a higher value in T1, T2 ratio maps, it doesn't mean that it has higher myelin, but it's just like a sensitive method, right? So I think that's the same. And I think the advantage of the T1, T2 ratio method over the susceptibility source separation is that it requires minimum post-processing. So for the susceptibility related or QSN susceptibility source separation, Chi separation, they all need tons of post-processing after the image has been acquired. And I think that's the real drawback when you want to use this kind of methods in actual clinical situation. I'll just leave it there.
Well, definitely in the clinical setting. So how long would it actually take to acquire and pre-process a pipeline
So
that
if
gives you a good separation?
Yeah, so for the original one with GRE and Spinnacle data and the iterative optimization based reconstruction, I would say the acquisition would take about about 15 to 20 minutes because like we have to acquire GRE data, which is about like 5 to 6 minutes, and then we have to acquire multi-echo Spinnacle data, but the multi-echo Spinnacle data takes more than like 10 minutes if we want to get decent max. So that would result in about like 15 or 20 minutes of acquisition. And then we would have to post-process it using iterative optimization, which would take about like 5 minutes or so using a standard normal computer. But the recent works, this one, the KaiSetNet, this one utilizes a deep learning based method to do that. And this one only utilizes GRE data. So using this one, we would say data acquisition for about like 6 minutes, and then post-processing, it takes only about a few seconds because it's deep learning.
That's an impressive improvement in time saving. Wow. Final question for you. So I'm totally biased towards inter-individual variability. So I was wondering how this method can handle inter-individual variability in iron and myelin distribution, but also how much variability there actually is in those measures across people.
I didn't get the last part, so could you just repeat the questions?
Sure. The question was how much variability there is in iron and myelin across people, or if no one has looked at it.
Oh, well, I don't remember the actual numbers, but there was this one. In this work, the normative iron and myelin paramagnetic and diamagnetic susceptibility map, he also reported the variability across subjects. So I think you can look into that one, this in around. Yeah.
Great.
I just
have a look
don't
at that one.
remember the actual numbers right
A pointer is good enough. Now I know where to look.
now. Okay.
That actually triggered a really basic question that I never thought about, but a lot of people, especially women, tend to be iron deficient. So I was wondering if iron deficiency is then also measurable in the brain, and if that was actually considered.
I think that's a really interesting question. Maybe I should like write a, what is it? Whatever. Maybe I should like start looking into that.
So I guess it hasn't been done yet. It just came to me. I'm like, that's interesting thought, because there's a lot of women, at least, that are affected by low iron, at least at some points in their life. So could be interesting to look at. Right. I don't see any more questions. So thank you so much. This was very exciting work to see. Thanks for joining us, and wishing you a good night after this talk. And down under, for everyone else, this was the last talk for the first half of the year. Now we're taking a summer break, and we'll be back in September. So see you next Monday for the last Neurochino, and otherwise have a lovely summer. See you in the autumn. Bye.